Hello, welcome to the next video from the conscientious biologist Ben Gallagher. This one's part of the A-level scheme. It's on saccharide biochemistry, and in this one we're going to look at glucose and glycosidic bonding. There's two videos in this playlist, and whilst they are part of the cells and ultrastructure overall playlist, which covers about half of year 12 material, these two are a bit more standalone because the saccharide biochemistry isn't particularly linked to any knowledge of anything else. As always, please do subscribe to my YouTube channel if you haven't done so already, like any videos that you found really useful, and you can follow me on both Facebook and Instagram. Thank you. So let's start off very basic with just the question, well, what are saccharides? Well, saccharides are just sugars, or more commonly we refer to them as carbohydrates. Now, when you think of carbohydrates, I would hope that you think of things like this: potato, rice, pasta, bread, fruit, vegetables, things like that. Those are all loaded with natural sugars, big chain carbohydrates that give you lots of your energy. But of course, we as humans learned long ago to process these natural carbohydrates and get the sugars out of them, and we can then use those sugars in more of the stuff that we all really like to eat, so like a chocolate cake surrounded by Kit Kats with lots of different sweets on top. That is much more refined sugars, but still carbohydrates. So everything on this bottom line here is carbohydrates. They're our main energy source as humans and indeed as animals. Now, you need to know about quite a few types of sugars. The first group is called monosaccharides. Mono meaning one, so we're talking single molecule sugars. Now, one of those should be jumping into your head from GCSE and prior study. There should be one monosaccharide that you absolutely know the name of, and it's glucose. You've studied it in great detail at GCSE and what it's for, and I would hope that you know that glucose is the main respiratory substrate, as in it's the thing that you use for respiration. It's the molecule that you get your energy from, so it's an energy source. What you won't know is that there's two main types of glucose. There's alpha glucose. That little symbol over there means alpha. It's the Greek letter. There's alpha glucose, which is the one we use for respiration. But there's also beta glucose. Now, I should have probably explained at the start of this slide, we're going to start putting in lots of overviews of the sugars, lots of basic knowledge. However, we won't fill everything in on this page because I'm going to put this exact page at the end of the second presentation when we can fill in the blanks. This is just kind of stuff that you should already know, or stuff I can quickly tell you. So you've got alpha glucose for respiration, beta glucose we'll talk about another time, and then you've got these two that you absolutely should know if you've watched the previous lessons and studied: deoxyribose and ribose. Both of those are sugars. Deoxyribose you came across as the main part in the middle of a nucleotide in DNA, and ribose is in the same position of a ribonucleotide in RNA. So you should have come across both of those. Two other ones that are quite key is fructose and galactose. Now, both of these are isomers of glucose. They're both C6H12O6, exactly the same with glucose, just their atoms are arranged in a slightly different way. You can use those two as respiratory substrates, so you can respire those to get the energy out of them, but more commonly, those two get converted into alpha glucose because alpha glucose is a more efficient respiratory substrate. But those are the monosaccharides you need to know. The only one I've left blank is beta glucose because we're going to get to that in the second video. Now, after monosaccharides, you have disaccharides. Now, disaccharides, as you imagine, are just two sugar molecules bonded together. So two of those monosaccharides stuck together will give you a disaccharide. There's only three that you need to know. You don't need to know what they do. You just need to know them by name and what they're made of. The first one of which is maltose, then sucrose, then lactose. Okay, maltose is when you get two alpha glucose molecules joined together. So if you've got an alpha glucose and an alpha glucose, you stick them together, that molecule is called maltose. It's one of the most common sugars we make. It's when we talk about malted grain, malt in cereal, Maltesers as a chocolate. Malt is maltose. It's the sugar that's in those things. Okay, sucrose is an alpha glucose and fructose, so the isomer. That's the most common fruit sugar. It's what plants tend to transport in their sap, in the xylem. So sucrose is another one. And lactose, lots of people are lactose intolerant, you may have heard of. That's alpha glucose and galactose, the two monosaccharides joined together. Lots of people can't process that because they don't have the enzyme lactase, or they have a version that doesn't work. So they can't break down the lactose, so it builds up. Bacteria have to digest it, releases a lot of gas, which is why people who are lactose intolerant quite often get bad wind if they do eat anything that's got lactose, because the bacterial digestion produces quite a lot of gases from it. Now, disaccharides then leads on to polysaccharides. Poly meaning many. We've come across that word before. Okay, there's a few polysaccharides you need to know, and you may already know most of these. The first one is amylose, glycogen, cellulose, chitin, and peptidoglycan and murine. Some of those should be familiar to you. You certainly should have heard of peptidoglycan / murine from when we did cell ultrastructure. Amylose, I would hope you've heard of from GCSE when you did digestion, because amylose is digested by amylase enzyme in the mouth, because amylose is just another name for starch. Okay, starch is also made up of something called amylopectin, so you've got amylose and amylopectin, but amylose is the main polysaccharide that you find in starch. We're going to talk about that in the second video. Glycogen should ring a bell with you, because when you think back to GCSE and control of blood sugar, insulin gets the body to convert glucose into glycogen. So glycogen's kind of like animal starch. It's almost the equivalent. Amylose and glycogen are what we're going to focus on in the second saccharide video, so we're not going to talk about those now. But the other three you ought to have come across, and you should know. Cellulose is the polysaccharide that makes up a plant cell wall. Chitin makes up a fungal cell wall, and peptidoglycan or murine makes up the bacterial cell wall. So lots of information on this page. Some of it you should know, some of it will be totally new, and I don't have a magic spell to help you learn all this. This is just stuff you need to memorize, because you could be directly asked in the exam, what is sucrose? Okay, and you've got to be able to name those specific monosaccharides. So take a screenshot of this for your notes, but it's just a case of memorizing it. Now, the most important one we're going to talk about is alpha glucose. And I said in the last one, alpha glucose, it's that little Greek letter for alpha. And you should know from GCSE stuff that alpha glucose, or ordinary glucose, has the formula C6H12O6. Now, because there's 6, you call it a hexose. Hex for 6, like a hexagon. You may remember that the deoxyribose and ribose in DNA are called pentoses, pent for 5, because they have 5 carbons. Glucose has 6 carbons, so it's a hexose sugar, C6H12O6. What you need to know is how those are arranged. So if I pull it up on the screen here, that's what you need to know. You could, probably won't be because it takes too long, but you could be asked to draw that in the exam, or they could give you most of it and ask you to fill in like the front and back or whatever. But you should, for good practice, know that structure. Now, looking at that, you might be going, oh, that's huge, I'm not going to get my head around that. I'm going to give you a really good process for learning it, that in about 3-4 minutes time, I would expect you to be able to just turn away from the screen, pause the screen, turn away, and you should be able to draw it out pretty much 100% correct. So what I would want you to do with this is remember some of the stuff that I told you in previous biochemistry lessons. Okay, the stuff when we did lipid biochemistry, DNA biochemistry, amino acid biochemistry. So carbon always has 4 bonds. Oxygen always has 2 bonds, and hydrogens always have 1 bond. And if you look at the glucose molecule, that's all it contains: carbons, oxygens, and hydrogens. And if you look at them, every single carbon has 4 bonds coming off it. Oxygens have 2. Not always obvious when you've got oxygen in the OH molecules. It's because the OH is the O has 1 bond to a carbon and 1 bond to its hydrogen with it. Hydrogens only ever have 1 bond. So try and remember that for all your biochemistry. But the process for remembering glucose: start off, draw a hexagon. It's a hexose sugar, so start with a hexagon. Then, this is a bit weird, but it helps. Turn it into a mouse with a tail and an eye. So if you look at the mouse I put up here, you can see its eye, you can see its tail kind of sticking up, eye sideways onto you. To turn your hexagon into a mouse, put a tail on the top and the eye on the top. So the flat bit on the top there, that's your tail and eye for a mouse kind of sideways on. I know it sounds stupid, but trust me, I've been teaching this for years. It does help. Okay, so get that. That's already your basic structure, because you look, the eye is the oxygen from over there. Then what I want you to do is put carbons on all the points. Okay, there's 6 carbons, remember, it's in the C6H12O6, but I want you to start putting them on clockwise from the oxygen. So over here on the tip of its nose, if you're thinking of it as a mouse, put them on every time there's a point or an intersection, and count them off. Really important to count them off. So 1, 2, 3, 4, 5, 6 on the tip of the tail. Okay, that's your main structure done. Everything else that you've got on here, oh, by the way, I'm just going to get rid of the original hexagon and just put the bonds in now, just to make it more clear when you're looking at it. You could leave it as the full hexagon, or when you've learnt it better, you can just automatically start drawing it like this. But everything else on here now are just filled with either H or OH. So you've just got to fill all that in. The oxygen's already got its 2 bonds, look. So all you're looking at is what's attached to all those carbons, and it's going to be H and OH. Okay, so firstly, complete the front and back carbons. Remember, that was the first carbon and the 4th carbon when we counted round. Now, to complete those, they need an H and an OH. H, remember, is hydrogen. So think high, like high and low. Hydrogen, put the hydrogens up high and the OH down low. Okay, hydrogen. Try and remember hydrogen. Okay, same on the back. The front and the back are exactly the same in alpha glucose. Then I want you to go to the 2nd carbon, so that's one almost immediately above me here, count round 1, 2. That copies the front and back, hydrogen, and the OH down low. The others, look, form a line, okay, the other 3 carbons, and I've always called that the naughty line, because they don't do what they're supposed to do. They don't follow the hydrogen, they flip it round. I sometimes refer to it as the opposite line. So with that one, their hydrogens go down low, and their OHs go up high. They're opposite, they're naughty. So on that bottom one, hydrogen goes down low, OH up. The next carbon up above it, look, which is the 5th carbon, hydrogen goes down low. It can't have an OH above because there's a carbon there, but that carbon could have an OH above. So then as your last step, step 7, you just fill in the hydrogens on the 6th carbon. If you count it all up around, you'll see you've currently got 10 hydrogens, so you just need the last 2 to fill out the 4 bonds of that. So 7 steps, hexagon, turn it into a mouse, carbons on the point, front and back with your hydrogen. Second one does the same, and then that naughty line does the opposite with the H's and OH's. If you can get that right, you'll get a big tick, full molecule completed. Pause the video, look away from it, and just see if you can draw it out. See how close you get now, okay? And if you can get pretty close, then it's easy to just make sure you've got it all the way complete. Make sure you've got this down there. It's a very important bit of biochemistry. Let's now look at how 2 glucose molecules bond together then to make a disaccharide, or then continuing on to make polysaccharides. Now remember, when we've done bonding before, we did amino acids bonding together and you get peptide bonds. We did a glycerol molecule bonding with its fatty acids to get ester bonds. The bonds that form between glucose molecules are glycosidic bonds. So if I pull up one glucose molecule there, and I'm going to put another one next to it that we're going to bond together. Now you should hopefully be at a stage in your biochemistry of looking at that and going, oh, that's going to be the same as the ester bond formation, because look, you've got 2 OHs next to each other, exactly like you did in ester bond formation, where you had the OHs from the glycerol and the OH from the carboxylic acid group of the fatty acid. And when you've got those next to each other, they're going to react together. They're going to lose H2O, lose those, so it's a condensation reaction to give off water. You've only got an oxygen left, so they bond together like that. It's exactly the same formation as we saw in ester bonding. The 2 OHs, you lose the 2 hydrogens and an oxygen. 1 oxygen's left, so I'll just put it in brown to mark it as clear. That in brown there, that is the glycosidic bond. Okay, you're very likely to be asked to draw a glycosidic bond or complete the glycosidic bond if they give you parts of the diagrams. Now, what I want to add to this, though, if you look at that glycosidic bond, you can see it's between the 1st carbon of the far glucose and the 4th carbon of the glucose slightly nearer to me. So that's called a 1,4 glycosidic bond, because it's between the 1st carbon of one and the 4th of the other. That's a 1,4 glycosidic bond. That's not the only type of glycosidic bond you can have, though. If I put this glucose molecule there, you can see that the top of it, the end of the tail, the 6th carbon, now puts its OH next to the OH of the 1st carbon of that other glucose there. So those 2 OHs could form a glycosidic bond. If they do that, exactly the same process, they lose H2O, there's an oxygen left over, so there we go, there's a new glycosidic bond. But that was between the 1st and the 6th carbon, so that's a 1,6 glycosidic bond. Now, if you look at it and try and imagine it on a bigger scale, 1,4 glycosidic bonds Literally, if you've got your little mouse, they're kind of going nose to tail, just making a straight line like that. Okay. In actuality, in the next video, you'll see that the line kind of curves around, but it makes a long chain. 1,6 glycosidic bonds, they're suddenly branching off at an angle. So 1,6 glycosidic bonds can make it branched, whereas 1,4 glycosidic bonds keeps it going in straight molecules. But to describe it, a glycosidic bond forms between two glucose molecules through a condensation reaction between OH groups on adjacent molecules, giving off water as it forms. That little description there ticks all the boxes, 3 or 4 key marking points in there. So take a screenshot of this, but absolutely make sure you can both draw and describe glycosidic bond formation. I'm stopping there on this video because that's quite a lot of information to have thrown at you. What we're going to look at in this next video, Saccharide Biochemistry 2, is how those monosaccharides can form disaccharides and then onto polysaccharides, and specifically with the 2 polysaccharides, amylose and glycogen, how the polysaccharide structure informs their function and why that's so important to the overall chemistry of the cell. So head to that one next, please. As always, do give this video a thumbs up if you've enjoyed it. Do subscribe to my YouTube channel if you haven't done so already, and you can follow The Conscientious Biologist on Facebook and Instagram for lots of supplemental material. Thank you.